for that wonderful introduction yesterday and today. It's been an absolute pleasure to be here at the University of Oregon and meet all of, I don't know, the amazing sort of graduate students and postdocs and even faculty that are here. So thanks very much for having me. It's been an absolute pleasure. When I was getting ready to develop this talk and introduce a lot of our oral microbiome transplant work, my graduate students were trying to help me come up with a new seminar title. And we decided on this from prehistory to precision, which I really appreciate. But they also did a chat version of this talk, which was entitled Neanderthal Oral Microbiome Extravaganza, Pioneering Transplants with Ancient Magic. So I thought a little joke to get us all started. So as is common of anthropology, we do usually start with an acknowledgement. And so I want to acknowledge that we have indigenous lands today. That the work done was done on indigenous lands of both Susquehannock and Ghana in Adelaide, Australia. So I want to pay respect to their elders past, present, and emerging. There are also human remains in this talk. And so if that is a problem for you, take care when we talk about ancient DNA and into that part of the discussion. So let's go ahead and get moving. What is the most chronic, widespread disease in the United States? Shout it out. Cavities. Okay, somebody knows, right? That usually isn't the disease that comes to our mind first. Usually maybe it's obesity, cardiovascular disease, or Alzheimer's. But it turns out it's indeed cavities. And in fact, 90% Americans experience at least one cavity in their lifetime. Huge disease. And other oral diseases are no different. And 70% of people over the 60, 70%, huge incidence of disease in the United States populations. And our primary way of defense against really common chronic diseases is to use toothbrushing. That's what we use as our sort of biggest line of defense. And the toothbrush was invented in the 1700s. The one that we see today in many people's bathrooms originated in 1938 with Dr. West's toothbrush. And it really doesn't, maybe from some toothbrush you might see in the store today. So that means that since the 1940s, essentially, with the same old technology to treat the most common chronic disease in the United States. And in fact, 50 Americans still use something that looks very similar to Dr. West's toothbrush. Half of us. In fact, who still uses a toothbrush that looks like that at home? Quite a few of us. Yeah. So we're still using a technology from 1938 to treat these chronic diseases. That might be a little bit of a problem. And the problem comes is that it really started to become mainstream. It was in a post-World War II era. It was in an era when antibiotics were being introduced. It was an era where things clean and destroy microbes. It's infectious disease. It's really an antimicrobial era. And to give you an illustration of this, that was given to soldiers enlisting World War II. And in fact, if you can see the things written on the chalkboard behind this man, give these sort of instructions to soldiers. The first instruction for these soldiers that was going to help them win the war was just to keep their teeth clean. Okay? It was a huge that the army thought was necessary for them to win the war. And so all ideology, all practice really stems from a World War II movement or a post-World War II movement where we needed to kill microbes and we needed to keep our teeth clean in order to and help. But I would argue that if 90% of us are suffering from oral disease, we probably need better preventive treatments for disease beyond toothbrushing and might even move beyond antimicrobial sort of rhetoric. And that's because microbial work. As I said last night, every surface you touch, every bit of air you take in, every sip of water you drink is full of microorganisms. Live in a microbial world. So why are we trying to them when they're all around us anyway? Now, the human body is different. In fact, every single person in this room, slide might look familiar from the talk last night. You'll see a little bit of overlap, but hopefully not too much. Every single person sitting in this room has 100 trillion bacterial cells. 100 trillion. If every one of those is a second, you go around, or if every one of those is a second, you still have over 3,000 sort of hours. A huge amount of diversity within the body. And over 50% of your cells in every single one of you is microbial. Over 50%, right? More than half the cells in your body are microbial. There are 1,000 species which is pretty amazing. I think when I try to think about 1,000 mammalians along those lines, it's almost impossible to do. So to think through 1,000 species in every single one of you on a microbial scale is pretty tremendous. And this equates for about 3 pounds of your total body weight, which is analogous to your brain. It's not just the fact that these species are present, though, but it's the genetic material that they bring with them. And it's all of the sort of the data and the function and the code in their genomes that's really, really critical. The microbiome in every single person contains somewhere between 2 and 5 million bacterial genes. Now, if the human has 30,000 genes, it means that 99% of the DNA within the human body is microbial. It blew me away because I thought, gosh, think about all the research that we've been doing using the human genome, and we've missed 99% of the information in the body. On top of that, that information is changeable. It's adaptable. Take probiotics, we can change that information. So if we think about new treatments, we'll be thinking about this other 9 of DNA in the body. And indeed, if we look to the mouth and we think about these microcavity causes of these most chronic common things like cavities and periodontal disease, they're also directly linked to things like oral cancers and bad breath, like halitosis. But these oral microbes are also linked to our systemic health. In fact, oral bugs, when they get out and go elsewhere in the body, are incredibly nasty and associated directly with gastrointestinal cancer or heart disease. But they can also be indirectly related to things like obesity, type 2 diabetes, as well as anxiety and Alzheimer's. So thinking about our oral microbiome is more than just thinking about our oral health. It's also thinking about our systemic health. And we know things like oral disease, like periodontal disease, or developing systemic diseases. About treating the mouth, not just for these very common chronic oral diseases, but also to improve our systemic health. So we really need treatments that leverage this microbial ecology and leverage this wealth of microbial information that's present within the body and present within the mouth. We really need to be thinking about this as we develop new preventative treatments. But how do we go about doing that? Well, this is where I think, and I argue, anthropology and archaeology have a really key role to play in this story, because we didn't always have lots of cavities. We didn't always have lots of periodontal disease. About wolves in the wild compared to your domesticated dog at home, do wolves have lots of periodontal disease and cavities? They absolutely don't. And the human the same, right? Human ancestors, for most places until agriculture came, had incredibly good oral. In fact, Neanderthals had excellent oral health, right? And so if we want to use that information to understand what our state of health should be, archaeology and anthropology have a role to play. To go back into time and to what a good oral microbiome look like and what these states might look like prior to this situation we're in today, where 90% of us experience a particular disease. Going back is by what I will call a time machine of using ancient DNA. Ancient DNA doesn't mean that it has to be super ancient. It just responds to the techniques, the DNA extraction and DNA library techniques that we use to get DNA out of a sample that was not intended for preservation. Okay, so when I say ancient DNA, I also mean historical DNA, degraded DNA, if we're applying those sort of techniques to it. There have been lots of these publishing ancient DNA recovered from soil, bone, feathers, hair, and eggshell, but it is also very difficult to work with. You can't get DNA out of things like amber, or very difficult to get. One to trust, but the other ones have pretty much been contamination. And DNA also has a half-life, and we would not expect to be able to recover DNA more than sort of 2 million years. So tens of millions of years is out of the question. We have DNA from dinosaurs that's reliable and reproducible, and Jurassic Park probably isn't real. But lucky for us, we do have the ability to go back in time, reconstruct oral microbiomes from people who lived in the past to understand what microbial communities looked like prior to disease, understand whether or not those microbiomes were associated with different states of health. And to do that, we use dental calculus, or preserved dental plaque. Now, this is a calcified film that grows, reproduces, and it accumulates over the life, much like tree rings accumulate with the growth of a tree. Excuse me. Allergies. Turns out when you come to holy smokes. Okay. Anyway, plus cement-like matrix, and it survives incredibly well in the archaeological record. In fact, it goes back millions of years to Australopithecines. You can see these sort of dental calculus reservoirs on their teeth, some different populations. But this is essentially a time capsule of the microbes that lived in people of the past. And we can go into different human populations as long as we have the consent of their descendant, their teeth, remove that dental calculus, and see inside as a way to sort of reconstruct the different types of microbes that people would have had in the past. But as I said before, working with ancient DNA is not easy. And in fact, working with ancient microbial DNA is even more difficult because we live in a microbial world. Microbes are everywhere. They contaminate most of the things to look at deep perspective. Also now ancient incredibly fragmented. We know that we have deep break down DNA into smaller and smaller fragments of time. So if we're reconstructing ancient microbial communities, we expect that DNA to be very, very small. And in some of the Neanderthals second, our average fragment length was 38 base pairs, okay? So modern DNA research, you know that that means we have millions more pieces of the puzzle to try to assemble, most folks working in modern contexts. We also know the DNA sequences cases. We've got a lot of DNA damage. We have deamination reactions that actually change some of the DNA sequences. So we say on the 5' ends of our DNA, and we need to account for that. We have taphonomy, different wall structures, and that means they're going to survive in the archaeological record at different rates. And so we expect some species to degrade at different rates compared to others. So we're going to have to account for that in all our analyses. And last but not least, we also have huge problems with contamination. Most of the animals we're working with have been in the ground. We know to contend with soil microbiomes or the microbiome of the decaying body, the necrobiome. We know we're going to have to sort of look at. We also have to contend with contamination on researchers, archaeologists, and museum curators. One of the most frustrating things about archaeology is archaeologists used to lick bones to determine whether or not something was a rock. And they introduced their oral microbiome onto these samples, right? And so we really have to be looking at oral microbiome of, say, an archaeologist from the 1920s, and that we're actually looking at, something that is real. So contamination is a really big issue for us. So we have spent the last 10 years working on improving these methodologies and being really, really that what we're looking at is indeed true or endogenous, and that we're not just looking at contamination and that we're not just looking at sort of junk in the environment. So in order to really go after this evolutionary history of the oral microbiome, one of the things we decided to do was do a longitudinal study in Europe and go back from when people were hunting and gathering and foraging in Europe all the way through to the Industrial Revolution. And so what we did was try to go after hunters and gatherers in Mesolithic Europe more than sort of 10 to 12,000 years ago. It turns out there's very few remains that correspond to anatomically modern humans in Europe. What there are more of, albeit still very small numbers, are Neanderthals. Neanderthals, because we know Neanderthals and humans are interbreeding before they sort of decline and go extinct from the world. Although I really argue, I think they just interbred with us, carrying as like European carrying Neanderthal DNA inside yourself. Right? So actively. Anyway, Neanderthals are a really great proxy for understanding what the microbiomes of Mesolithic and Paleolithic humans would have looked like. And we compared these to wild-caught chimpanzees. It's important that they're caught, because if you take zoo chimpanzees, they have a very different microbiome from that sort of zoo environment. So we have wild-caught chimpanzees, and we compare those to modern humans to just get an initial reading on where we are in a modern world, right? These are folks living in industrialized communities, obviously in Adelaide, Australia. Most folks are of European descent. One of the things we saw straight away, though, kind of blew my mind. I was expecting for there to be species differences. I was not expecting to see large-scale differences at the phyla level. What we see, though, is that there are lots of blue pieces of the pie here in chimpanzees and Neanderthals, and those correspond to gram-positive species. Most of these are streptococci, for microbes to bind the teeth, and are often associated with a protective sort of phenotype. Gram-negatives, this includes things like Porphyromonas gingivalis, which we know can be associated with periodontal disease, and gram-negative taxa in their mouths. And that was quite shocking to us. We're also really surprised, too, to see a lot of this Neanderthal. Okay? A lot of that corresponds to a taxa called Methanobrevibacter oralis, and we've been chasing this for a long time. But what you can appreciate is that there's sort of unique species and many more gram-positives in Neanderthals and chimpanzees, and that Neanderthals and chimpanzees look much more similar to each other than, say, people living today. So again, suggesting folks living today probably have a sort of dysbiosed or an altered microbiome, which see high incidence of disease. We did a quick comparison to other humans through time in Europe, but these do range from different geographies in different contexts. But some of the things that we found that, one we found, sorry if I can get hold of my pointer here, is that we do see a shift in the microbiome between people who are either foraging and hunting. We do think that hunting probably plays a role in some of these first shifts that we see in the European microbiome. Second we saw was when we started using agriculture. In fact, we do see a shift here. This has been repeated by several different studies to show that the introduction of agriculture across Europe in different settings really does shift the types of microbes that we see in the mouth. And this is associated with higher incidence of oral disease, things like periodontitis and cavities. The really is actually Fusobacterium, which starts to come in about this time period. It's shown there. It's one of the red colors. And this species is the keystone species in this ecology. It really allows this microbial community to build much larger structures. And we might actually think that that's why we see increased incidence of things like periodontal disease, which is directly tied to the building of these really complex structures in gum tissue and the tooth. Really exciting through time. But as I said before, a lot of these folks are coming from different ecologies. The sample size here is incredibly small. We're really, really limited. So even though this is one of the first studies to ever sequencing to ancient humans and was the first study to reconstruct an extinct microbiome, one from an extinct species, Neanderthals, there was a lot of work left to do. And so we set out trying to get much better resolution in many different around the world, doing this on every single continent, and trying to increase our sample size, do much better community collaboration, and also really get a handle on what are the local features and factors that might have altered the microbiome. We were really interested in doing a transect in the UK or in Great Britain because we thought that this might actually be a good place to understand the birthplace of industrialization and understand how industrialization influenced the microbiome. And so indeed, that's what we published this in Nature Micro, and it was a huge, tremendous effort from both Andrew Farrer, who's now at the Cambridge Science Center as a lead science community, Abby Gans, who's in my lab at Penn State, and I'm going to postdoc at NC here in a few weeks, actually. Anyway, the last 2,500 years in Great Britain, and it's included 127 well-contextualized individuals from London. Sort of see this peak here in the medieval. Those folks are incredibly important, and I'll talk about them more in a second because they have really, really good archaeological data that comes along with them. They've been studied for decades at this stage, and so we know exactly what sort of skeletal diseases they might have suffered from. Some of them we even have nameplates, know exactly who they were, when they lived, a lot about them. So they become very, very important in understanding what actually shifts the microbiome in the past. One of the shocking things, though, when we put this together, is that we observed this sort of U shape on this PCOA plot. And it tells us that we have very distinct ecologies. We've got very different things going on. And so we started to try to understand just straight away before we started testing anything, what is driving this massive sort of U shape? And it turns out that we have two very different microbial ecologies that are largely driven by dominant taxa present in either one of those ecologies. So on the right, you have these green, these are rural microbial communities that are dominated by Streptococcus species. And it turns out this is what we see in industrialized folks today, is this Streptococcus. If you look in a dental textbook, you'll see this community described in the dental textbook as the oral microbiome. What we did not expect were a bunch of oral microbial communities that are dominated by Methanobrevibacter oralis. And in fact, this archaea was driving an entire microbial ecology that was completely distinct from the ecology that we saw in Streptococcus-based communities. So it's not just the presence of one dominant taxa, entirely different microbial ecology we're not aware existed in the human mouth at all. Okay? Now, it doesn't mean it's absent from modern populations today. It's really that they're incredibly low levels. And it means that it may have linked in some contexts that we might compare in the past. There are also lots of other different ecologies too that are dominated by like actinomyces or prevotelles, but they're much more, I would say, just in sort of what their dominant attacks are. So really, really surprising to find a new ecology that we didn't existed in the past. We then set out to understand what the heck was driving these different ecologies. And we had all sorts of questions that we wanted to ask history of the UK might have shaped the oral microbiome there. We thought for sure that we would see sort of evidence of things like physiology, like age, playing major cultural or religious transitions. We might have seen sort of shifts in the microbiome. We also looked at Asian came to the, and we thought for sure we would see some French microbes, coming into the British. Turns out we don't see any of that. All of our hypotheses were wrong. Okay, that's a frustrating day, right? I should say that they're wrong. All of our suppositions, predictions were wrong. The only thing that turned out to be associated with any sort of difference between these two ecologies was actually time period. You zoom in, change occurs. It actually occurs in the 1600s, which happens to correspond with the arrival of the second plague pandemic in the UK. Now, called the Black Death, we try not to call it that for sort of respect reasons, I would say, out of the people who passed away. So we call it the second plague. And it's a time period, at least in London, where somewhere between 30 to 50% of the population passed away, right? So an incredibly devastating pandemic. Now you might think, well, maybe the microbiome just changed because there's factors that select for survival, right? Maybe the people who survived have a different style, have a different microbiome that helps protect them against infection, maybe something along those lines. And you might be right, you might be completely right. But we really are lacking tools to be able to ask many of those questions. But one of the very interesting things that happens after the second plague pandemic are drastic changes in things like hygiene and cultural practices and so forth. And what's been reported is actually just sort of improvements in life that happened after this plague pandemic. You had societies that were set up, and now you're missing half of the people. And so it allowed workers to request much higher wages. They asked for better food, working. This has shown dramatic increases in the amount of meat consumed at that time period because workers were able to have better meals, access. So one of the things we set out to test actually whether or not diet might have driven this difference in the UK. And you can do a lot of research to try to pull out dietary DNA. You actually do have little bits of food stuck between your teeth. It's a tremendous work, and I will say microbial DNA contamination and eukaryotic reference genomes and NCBI is there. But we did a different approach that would be more reproducible. And that's actually to look at the microbial functions in these sort of organisms that we're seeing and see whether or not those microbial functions are tied to known dietary inputs. And so what we did was look at different functions that are known to be associated across the mammalian kingdom, like meat consumption, milk consumption, or even sugar consumption, or even diets that are high in sort of plants and high and low in fiber. One of the things we saw that demonstrated two separate microbial communities was that folks here with the Methanobrevibacter community shown here on the left, and folks with different functions associated with the Streptococcus community shown on the right. Things that we see is that the Streptococcus community actually has functions enriched for things associated with things like dairy and sugar, okay? So has things over here, or things, functions enriched, I've really been talking too much, as also, and so that suggests that people with the Streptococcus microbiome, the one found today in industrialized settings, was associated with a diet that was probably richer in things like meat, dairy, and sugar. It's like you can also see dairy proteins hidden in calculus. It matches what we expect to see. We also know that there's a huge influx of sugar about this time period, right, with increasing international trade coming into the UK. So it matches what we expect to see from the archaeological and historic records. But it also shift to a richer diet and shift to a diet that's more akin to things today, industrial settings, may have also helped select for the types of microbes that we know are in people's mouths today. So that might be really, really great. And we could, if thinking about all this improving health and modern today, we might say, okay, that's great. Well, maybe just move over to the Brevibacter community. We should just try to shift everybody over to a community that's associated with that archaea and get away from this community today that we know is associated with problems. Actually, then not either one of these communities was associated with health in the archaeological record, because we can see signs of disease in the skeleton or in the bones of those people, also track an oral and different things. And what we found was the Brevibacter community was not oral health issues, and so that was really but it was associated with other systemic health issues, okay? So in fact, Neanderthals associated with arthritis, ossified lipping, and porosity of the bone. Arthritis in that sort of context. So it might be that even though we're selecting for microbes that might improve or microbes that actually cause other diseases. And so this might not be that clean cut. It might be much more difficult to figure that out. So we in the UK and we published that. We're now working in all these other different contexts and working on the sort of evolutionary histories that occur around the world. Really excited to publish is one on the western coast of South America, working with communities in both southern Peru as well as northern Chile. And we've been comparing the types of microbial communities that we see in people who lived in South America at the exact same time that these folks living in Europe. And striking things we're seeing so far is that we see incredibly unique microbial ecologies, the same people in the UK at the same time period, okay? And this suggests to us that each of these evolutionary stories in different populations are going to be different, and that different communities, especially to the colonial era, had different microbes in their mouths, right? And they also had different microbes that are associated with disease. So we're continuing to share it with you, hopefully next year. it'll be an exciting ride. But it means that we need to go back and redo evolutionary in each and every single population that we're interested in studying, and not do them sort of on these continental, sort of wide-based approaches. So I'm going to wrap up sort of our ancient DNA stuff, move into two other areas of talk, but at least the ancient DNA, I hope you're going to take home that, you know, ancient DNA once had an oral microbiome with systemic health, but it was linked to oral health. Social and cultural transitions did select for disease-associated microbes, and that was probably largely through diet, including the introduction of large meat and milk into our diets on a daily basis. And last but not least, evolutionary history of our oral microbiomes is going to be population specific. And if we want to get this right, we're going to have to think about what the evolutionary history of the oral microbiome is like across all people and in different locations. But if we wanted to take that information and sort of do something with it, what might we do? Well, we can't really use this Methanobrevibacter community because we know it might have ramifications for systemic health. But we might be able to look at people today who've never suffered from oral disease. I told you earlier in the talk that 90% of people have a cavity, the other 10% who've never had that, right? They may actually be the key to sort of thinking of how microbial ecology can come to rescue some of our diseases and think about new therapies and ways of treating disease, leverage microbial ecology. So there's a couple different ways to think about this. One by using sort of probiotics. You know, most of you probably thought about this in the context of yogurt or kimchi, or maybe in some of these designs they're coming on board now. That might, by putting single species into a whole new ecology, they often don't survive, not sort of long-lasting effect. You might also think about prebiotics. You might think about sort of feeding food to some beneficial microbes. We might be trying to encourage the growth of those gram-positives while sort of limiting the growth of those microbes associated with parasites. You might also think about phage therapy, right? Having specific targeted viruses that go to keystone species that sort of deflate the biofilm formation. People have tried it. It doesn't work. Okay, that biofilm is incredibly robust to any sort of phage getting into it. Also incredibly robust to things like antibiotics. Really, really frustrating. And last but not least, you might think about oral microbiome transplant. Now, this has been done in the fecal world or in the gut world for a long time. Fecal microbiome transplants have been something that's now almost common knowledge, which is bizarre to me, right? Where you actually take microbes from a donor and you sort of whiz them under some saline solution, plant them into a recipient who might need them, okay? For an entire ecology that's well suited to sitting up together in that individual. But this hasn't been developed yet for the mouth. There are a couple of theoretical papers how it might be able to work, but it hadn't yet been tried. And so about 5 years ago, I wrote a grant to do just that, to see if we could. I've been working with some really awesome dentists and oral microbiologists from the University of Adelaide and so PhD and Kevin Kitagoda to really figure out a way to develop what we call OMT, or oral microbiome transplantation. Now, the premise here is very simple, right? We know that we have diseases that are caused by the microbiome. And so if we can sort of obliterate somebody's microbiome, We can then take plaque from donor. We can grow that up in lab setting to ensure that it's safe, doesn't have infectious disease, viruses, or other pathogens we might not want to implant. And then we can mix that with a hydrogel and essentially paint that on the surface of the teeth donor, or even put that in a sort of gel whitening tray, put that gel in a whitening tray and sort of put that, tray on your teeth, just like if you were whitening your teeth. Okay. So it's not exactly rocket science, but the premise is there. We started off, though, by trying to figure out who the heck we might use as donors. Who is that 10% that we're after? Who are those sort of magical unicorns of people who have never suffered a cavity? I'm sure there's some of you sitting in this room right now. And so what we did was recruit 100 what we'll call ultra healthy folks. And so these are people who either have never had a cavity or maybe who've had cavities, but it was at least sort of 10 years ago. And they don't have any signs of gum disease in their mouths. They've also had no incidence of oral cancer or any sort of other oral health issue. Now, we were told by many dentists that this will probably be a very strange proportion of the population. It'll probably be the people who are vegans and only eat leafy greens and probably run marathons and are like these just strange, ultra healthy people. Turns out that's not true. In fact, a lot of our really interesting ultra healthy donors are people who eat tons of sugar a day, right? They're not marathon runners. A couple of them smoke occasionally. We exclude that, right? These are not folks that you would have expected to have incredibly good oral health, right? But indeed they do, okay? So what we did was collect anybody who sort of fit our criteria. And in fact, I will say our best data on here in a second is who eats at least 300 grams of sugar a day, right? But still never had a cavity, right? These are the sort of unicorn type people that we are after, okay? These are our donors. There's actually an incredible amount of diversity, an incredible amount of factors, healthy, ultra healthy oral microbiomes, okay? Things like how often you brush or how often you floss play a key role. Your pH and how much sugar and all sorts of things that you do can play a role here. I will say, though, we pretty much do have sort of two populations. We have a population of people who never brush, never been to the dentist. And we have a population of people who are probably in dentistry school and we might have outside of one of those. But they're people who brush their teeth times a day, they floss every day, and they take incredibly good care of their teeth, right? And that sort of oral hygiene effect there is probably one of the biggest declines that we see. We then took these donors and we developed a 3D printed cell to essentially mimic the human mouth, dental plaque in this in vitro flow cell. This is actually artificial salivary media, which we pipe through this pump into the flow cell. And then there's this sort of, export sort of tank here. And in this, we have this port here where we can in sort of donor dental plaque, which seed grows on these discs, are hydroxyapatite, which is essentially human enamel, okay? And with that flow, and in that sort of V shape, with the sort of enamel discs that mimics the human mouth, we're able to sort of grow microbiota in this 3D printed in vitro flow cell. And we can also parallelize this and grow lots of donors all at the same time. We've assessed these donors using microscopy to make sure we can grow these in vitro biofilms, to a really high standard. In fact, this is growth after 14 days, and we'll see we still have upwards of 75% of sort of live cells still growing in there. So that's really, really exciting. You can also appreciate that even by looking at this using scanning electron microscopy, they have different oral microbiomes, okay? They don't all look exactly the same. And in fact, if we look at 3 of those first donors, RNA sequencing, we can see that they have individuality despite being grown in vitro, which was a huge question for us. So here we have collected or inoculated plaque, both shown here and here. And here's the growth in the flow cell after that. So we do see a small reduction in diversity, and we lose a lot of the strict anaerobes that are present in that biofilm. We are able to maintain individuality of the microbiomes, which is actually really exciting for us because it means we have lots of opportunities to access many different types of donors and sort of harness this diversity of the microbiome in different ways. Now that we were able to sort of throw up donor plaque microbiomes, we then wanted to sort of transplant them and see whether or not they can actually be used to prevent oral disease. And so here we're using a Sprague Dawley rat model. This is one of the only sort of caries development models of this. And what we did, young rat, can either give them an OMT when they were receiving diet. And so basically these rats are fed high amounts of sugar in order to sort of induce caries. We also made sure they didn't have rat forms of streptococcus that can cause caries. In some of the cages, we actually infected them with Streptococcus mutans, which is a microbe that's known to be associated with caries and can produce high amounts of acid in the mouth. Half the mice, though, despite that carcinogenic diet and whether or not they were given that sort of food, we then gave them an oral microbiome transplantation. And this was done once a day for 3 days by painting this microbiome mixed hydrogel on the rat's teeth when they're exercised. And then after this sort of washout period for 18 days, we were able to sort of look at OMT success, a wide range of imaging, and also assess safety. Although we're still processing a lot of the safety data, though I can tell you it looks really, really good. And I'll show you just this clip of some of the results because it's still ongoing and still coming out as quickly as we can. And we're really, really excited about the future of this. In this rat model, just the control hydrogel, or once given hydrogel plus strep mutants, developed a higher amount of caries than either groups of the mice that were given an oral microbiome transplant. In this group here that was not given strep mutants with OMT, we saw caries be reduced by 56%. 56%. Some of the best technologies on the market today reduced by. So this is really exciting for us. It's preliminary evidence. We've got a lot of work to do. We've got a long road to go. We're really, really excited about this effect. We were able to introduce microbes into a rat and prevent caries formation in many of these rats. Super, super exciting. So with this work, developing oral microbiome transplantation, we think it's a really promising new technology to treat caries. And in fact, future work this summer will test it in a periodontal disease model in mice. So really, really excited where that might lead. We've also only tested this in 2 donors. We have 90. So hopefully the NIH will provide us something here shortly. But growing dental plaque in vitro also creates this awesome new system to be able to understand how oral communities, how we might manipulate them, and how we might understand to health in unique ways. But like I said, it's just the beginning of the road, and a lot of ongoing research is going to need to test its efficiency in lots of different folks, and also make sure it's incredibly safe. And I will say our early preliminary health data or safety data has been incredibly encouraging. We haven't seen any sort of signs of infection, and which is really true. But with research, I would say many more questions remain that have been answered. That's what I find happen in my career a lot. And so we're still really wondering about whether one's safe. We really want to make sure it's safe before we move into humans. We're also really curious to know whether or not OMT will permanently alter someone's oral microbiome, or whether or not it's a very short-lived effect, sort of efficacy of transferring microbes and seeing how long they'll stay. And what are the long-term health consequences of that? If we shift someone, will then be systemic disease? We don't know yet, but we need to find out. We also want to know how we make OMT more accessible, affordable. If we're developing a technology that can help address health disparity, we really need to make sure that everybody has access to it. It's not something that's incredibly expensive and hard to get a hold of. We need to make sure it's effective in all people, okay? I was talking to you earlier about how people's evolutionary histories and their microbiomes are different. How does this work in people who have different types of original microbiomes, the recipient microbiomes? And I want to spend the last 30 minutes of this talk focusing on the question, will OMT be effective in all populations? And this is where technology has a role to play. So we've been doing work collating as many sort of industrial as well as non-industrial oral microbiomes that we can find published today. We've added this with some data that we have from Uganda populations with the Batwa and the Bachiga, but you also find things like the Yanomami in there and other rural populations in Africa. And we've got European and American data here. That's the same as the gut. Industrialization is the single biggest factor that drives oral microbiome diversity on the planet today. But this also means that if we develop an OMT treatment communities, it may not work in other communities that don't have that sort of industrialized type microbiome. And so we've got to be really careful about who we're inviting, to come in and to provide their sort of microbes, because we probably need to be very much aware of a large gap in oral microbiome diversity around the globe, and maybe thinking about sort of matching sort of donors with recipients in some way that with their ethnicity or their. We've also been trying to understand whether or not specific microbiomes that are unique or separate from industrial microbiomes can be associated disease in ways that we didn't expect. And we've been working a lot with Lisa Jameson and in both South Australia as well as folks in Bamaga and in Northern Australia, Far North Queensland, to try to understand whether or not indigenous biomes that are very unique from what we see in industrialized sort of European populations, whether or not we could even rescue those with OMT. But we've been starting that project by looking at folks who are suffering from periodontal disease. And in fact, in folks who have severe perio, we see that Indigenous Australians have a very different microbiome profile, increases in alpha diversity or the types and numbers of taxa that are present compared to non-Indigenous folks. And in fact, if we look at the composition, we also see that from periodontal disease have a very distinct oral microbial community compared to the people of descent. And so again, thinking about some microbiology we're trying to replace and making sure we're not replacing that can then cause disease in some other unique way becomes really, really critical to OMT efficacy, especially in different populations. And I will say, I talked about this a little bit last night, but it's one of my sort of favorite stories about some of the work that we've done. In Aboriginal Australians, we also find microbes that we had no idea to get them out. We're something that we need to be thinking about in the context of OMT. In fact, Aboriginal Australians from the Central Desert have a species called microbium that's really reproducibly only Central Desert Aboriginal Australian populations. And microbium otherwise is only found in the guts of termites. Okay, this is the microbe that allows termites to get energy out of cellulose and digest wood. So eating termites is a very common practice in the Central Desert. It isn't really today anymore with industrialized food sources, but in the past it was incredibly common. And so it makes a lot of sense to understand how termite microbes might have gotten into the mouths of Central Desert Aboriginal Australians. But the fact that they're being maintained across generations and that they're still present in people's mouths and now associated with things like periodontal disease means that we have a lot of work to do to understand how unique microbes that we didn't even expect were going to be there are contributing to disease in ways that we couldn't have predicted. So we are working with many different populations around the world, with folks in Kenya and Canada, South Australia, and even Brazil with the Shavante, to really ask questions about how health is shifting in their communities and what sort of microbes are associated with oral disease in ways that we wouldn't have predicted in our analysis. If we're thinking about this in sort of an anthropological way, we really think that using industrialized oral microbiota are on to result in effective disease prevention across all populations. It really means we're going to need to include all people in OMT research and make sure that we're selecting this pool of sort of donors where we consider the cultural, historic diversity, genetic diversity of. That also means we need to think about how we compensate potentially in very different ways. Compensation for donors in Indigenous communities is very different from compensation in industrialized population. And we really think that personalized or dentistry approach, even matching a donor to someone or matching a recipient to a donor where they share the same heritage, they share the same culture, share the same history, might be a way. But we're going to need to make sure we ensure equitable access to the technology to ensure that everybody has a fair go. So I've told you three very different research areas in my team today, but I hope when you leave, I hope you're thinking about how leveraging microbial ecology might open new doors for oral health therapies that we otherwise thought about and that don't exist today. Archaeology and anthropology are going to have a key role to play if we want the technology to be accessed by all and effective in all people. And last but not least, thinking about how we develop and commercialize this technology to ensure that it's equitably accessed is going to be really, really important. And it's something I have a lot of sleepless nights about right now. So with that, thank you so much. It's been an absolute pleasure to be here, and I look forward to all of your questions. Thank you.
Laura, I might have you briefly restate the question just for folks on Zoom.
Sure.
Cut out very meat and sugar after watching this talk.
Yeah, it's a great question. So the question is how stable the oral microbiome. So If you look at, say, the gut microbiome, and the skin microbiome, and the oral microbiome, oral one is the most stable of any of those sites in the body across an individual doing longitudinal. No comparative, very stable. Attributed to the fact that it's a biofilm that forms in the teeth. Selection of microbes can work together is much stronger than, say, adapting to slight nutrient changes. All of that said, you can shift the microbiome, right, by shifting diet. There's a lot of work, for example, looking at intermittent fasting right now to see whether or not that shifts oral microbiota communities, and it does. And we know, for example, rates of nitrates, for example, can shift, slight variation in the microbiome. It's not going to shift it, as dramatically as what we see in the gut, where, you're just depending on the meal and what you ate shifts all over the place, right? The oral microbiome is stable, and it's much more stable, and only slightly changes with some diet. So there's been a lot of push for OMT technology in the dentistry field because of the stability of the oral microbiome. People said, well, there must be host selective factors. There must be reasons that don't allow an oral microbiome to change. And I would argue that's true, right? It's incredibly stable. But if we can move biofilm and replace the biofilm, and we can get the biofilm to seed and be, set in place, we might have a much better or more likely chance of being successful. As fecal microbiome transplants have been in some folks.
Great. Questions. One. So firstly, you showed that the huge population. Or I think we're going to keep that more information about how that a lot of the absence of happening. We get that. So I think when you maintain the regional diversity.
Those are some great questions. Okay, so the first question is about archaea, right? Like Neanderthals and other what I call ancient humans. It's even through into the Industrial Revolution, right? In places like UK, although we do see it sort of decline over time. And so there's a lot of folks really interested in methanogens in the mouth because they've been very poorly studied and very poorly characterized. Primers we use a lot in oral microbiome research. We have looked for methanogens and archaea in our populations, like Aboriginal Australian populations. We don't see them. And so why that is, why there might be geographic structuring to the presence of this methanogen is really fascinating to us. There's also been some work done in Portugal and in Spain, sort of looking at methanogens in the mouth and the saliva, and they can find methanogens, oral methanogens in saliva of an individual, and that same individual can't find them in the plaque. And so now it becomes kind of curious as to where these bugs are actually living. Are they living in some other soft in the mouth, or what's actually the deal there? So I'd say there's still just a lot unknown about oral methanogens and oral archaea in general, and we're really keen to work with some of those teams. I have a Zoom call when I get home to actually try to set up some future grants to try to where have the archaea gone, or are they still around? David Bellman had a really beautiful study sort of looking at archaea in some folks with severe periodontal disease, and we do see it in about sort of 20%-ish of people who have severe perio. They still have some archaea, but it's not nearly to the extent that we see it in the archaeological record. So, and there's been really cool ancient DNA studies looking at this archaea diversity in ancient populations. We showed that archaea were swapped between Neanderthals and humans in another study we've done. So there's all sorts of cool stuff going with archaea in ancient populations, but it's really not absent, but really at low levels in modern populations. So what the heck's going on, I think, is still a really outstanding for all of us. Yeah, and your second question was just thinking about like, well, first part of that question I'm gonna answer is just thinking about culture health, absence of straight away, our old folks was going presence of Streptococcus mutans. Now, in the dentistry literature, this is the microbe that produced lots of have cavities. And that's true to some extent, although we know now with microbiome literature that there's lots of taxa that can help contribute to causing cavities and severe cavities. I think, and lactobacilli can also contribute to that sort of high acid environment in the mouth. What we're using to sort of mineralize the enamel. We did find in about 20% of our ultra healthy donors, and so the sequence of 16S sequences, and I've got shock data coming down the pipe here very shortly. And it is the interesting part. Through the archaeological record, we've been trying to reconstruct Streptococcus species. We know that ancient rocks very rarely had strep mutants. It's very rare in the archaeological record, but there's lots of other Streptococcus species records. So we know by looking at some indigenous folks today that things like Streptococcus can cause cavities and have associated with high acid production. So it's the presence of the pathogen, it's the presence, the ability of function to do whatever it is that causes that disease, right? In the case of here, produce acid. And so it might be that in these ultra healthy donors, even if they have strep mutants, might be variants of strep mutants that don't produce acid, or they might be part of other factors that can buffer that acid and keep it sort of keep it at bay. So we've been trying to track the pH of these biofilms and really look at that. And in fact, in our first OMT, we selected donors that had really storing capacity in order to try to sort of balance out that acid produced by strep mutants. So yeah, I think there's a lot to also pathogen creation. And in the oral microbiome community or in the dentistry community, we talk about not pathogens, but pathobionts. Because there's a lot of people who have what we call the red 3, the microbes that are associated with periodontal disease. They have them in the mouths, but they don't have periodontal disease. And so, what about the ecology? Those pathobionts grow to really levels and then cause disease. But there's something that happens first that limits the control of those pathogens. And that target with OT is the control of potential pathogens in the mouth. So, and then your last part of the question was just about whether or not sort of that regional specific structure might be maintained. And it's a great question, and I don't know the answer. And I'm really keen to see what this looks like across diverse populations as well, right? And so we're really keen to talk to Aboriginal communities and talk to other Indigenous folks that we work with to see whether or not they would be willing to embark in sort of OMT research for that community, right? And some of these groups that have very different microbiomes, is that something that if you were a European person, does that stay or does it? Or is it that they're not the right sort of set of microbes? We don't know the answer to that yet, but I hope we will know the answer in the next few years. Yeah, great questions.
Thanks again. I was curious when you're looking at like systemic, why diseases you can like distinguish effects of like whether positive body.
Yeah, good question. Are these things so the question really like, is the oral microbiome associative or causative, right, with systemic disease? And so there's been some work in some diseases that's been really, really awesome and very mechanistic. So colon cancer, I would say, is the best example. And so Fusobacterium nucleatum, that species that is the bridging keystone species, if it gets loose and gets into the gut, it is really great at binding all sorts of other microbes and sort of microbes and causing colon cancer. And so there's been really awesome microscopy work and DNA work and cellular biology and done in suffer from colon cancer. The presence of this oral microbiome or this oral microbe getting in there and causing problems, right? And so very sort of microbes get elsewhere. There's also been a lot of really good cardiovascular research that's looked at strep species binding heart tissue and oral microbe plaque and things like that, and that to what they see in somebody's mouth, and they're not present, say, in the gut or somewhere else. A really cool one that's emerged, also right? These gingipanes, these proteins produced by Porphyromonas gingivalis are being found in sort of plaque tissue, right? For all, there are things truly, that all signs point to something, right? Whereas other diseases are very much associative, right? And so like type 2 diabetes, obesity, there's clear markers in the mouth for those diseases, but is oral microbiome causing obesity? Like probably not, with other things going on. So it just depends on the disease. I hate that answer, it depends, but as we were talking about last night, but, that probably is which disease. Yeah. Yeah. Some of the we're looking at, for example, some of the parakeets, which we know can be associated with things like periodontal disease. And so we've been really, we're like, we probably don't want those anyway. So we've lost them because you can still get a healthy plaque, biofilm them would probably increase your risk at maybe developing microbiome or mixed microbiome with their microbes or something. So we haven't been too concerned about that yet. But I think thinking about, right, it's in their mouth and the recipient, and we the recipient microbiome, right? We do scaling and rats anyways, and we don't have full scaling, and then we do a complexity and wash to fully sort of kill everything out. But if those, just like when you go to the dentist and you get that cleaning, your microbes come back. Some donor microbiome that's been put in, right? Maybe you do get those anaerobes. Don't. Really, like we've been thinking about it as like they're on when we put them in, but are they still lurking and hanging out and coming back in some other way? But only time as we start to sort of move this into humans. Question. Yeah. No.
That's okay. Lots of questions. Good.
Easy with. I'm wondering how challenging is it when you're doing shotgun sequencing and you have these sequences from ancient microbes. Someone kind of asked about this last night about like microbial evolution, and you're comparing this to a reference species. So even modern microbes, like is that a problem?
Yeah, it's a great question. We spent a lot of time thinking about this because when we do shotgun sequencing, we can only identify somewhere between 50 and 60% of the sequences we get out, right? Which is annoying as a piece because it makes it really expensive to just throw out half of your DNA, right? And so we've tried some de novo assembly techniques. They are very, very poor because our fragments are really, really short. And there have been sort of studies where we've tried to take, and we've done this actually in French Polynesia, where we've taken the 16S sequences out of that shotgun data, used that to build a sort of reference library to then map, and then come back around to sort of improve our mapping. And that improves things a bit, but not sort of tremendously. And so for us, it was really, really frustrating. We wanted to know whether or not we were like missing half of the microbial diversity that was just present because we were throwing out all these sequences. And so we did a lot of work to sort of enrich or pull down all of the 16S sequences that would be present in our samples. And 16S is incredibly well conserved. As you know, it's why we can use it as sort of a marker in our analyses. And we were able to classify all the 16S sequences we pulled down. So there were no sort of unique clade of 16S taxa that corresponded to some species that had never been described, or some genera or something, definitely species, but not like whole families or something like that that had never been described of microbes. So we feel pretty confident that we're able to, I would say taxonomically classify what's there. Functionally, though, it becomes quite frustrating, right? Because we know we're probably missing a lot of genomic diversity in these regions, or in these areas where we have gaps, right? And we have that we know we're not reconstructing correctly because we have to modern reference to do a lot of that work. So yeah, I would say de novo assembly is coming along with some of these short reads, new techniques, but it's still painful for us. It's very, very painful. So yeah, I'm hoping to improve on that. We've been working a lot on improving our functional databases and really trying to get a better handle on identifying some of those unknown sequences. But it's been rough. It's very rough. And so I worry about some of our functional analyses right now, whether or not they're, are we missing half of what's there, right? We spent a lot of time thinking about that, losing sleep about it, but anyway, so I think it's a problem as much as taxonomic identification. But today, I think it's a bigger problem in functional identifications. Yeah. Yeah.
I think we can put it there for now. Let's thank Laurel one more time.